Hi everybody. Today we discuss about animal biotechnology course under which we discuss unit 1 animal breeding. First we see definition for animal biotechnology. Animal biotechnology is a fusion of scientific and engineering principles to the processing or production of materials by animals to provide goods and services to humankind. Animal biotechnology application includes livestock, poultry, fish, insects, companion animals and laboratory animals. What is breed? A breed is a particular group of animals having identical or homogeneous phenotype, behavior and other traits that distinguish it from other animals of the same species. Examples of certain breeds. Under cattle, there are few indigenous and exotic breeds. Under exotic breed, Angus, Austrian Chabre, Chianiana, and Cole Jersey. Under sheep breed, Boer is the South African breed. Coming to indigenous breed, Tarparker, Red Sindhi, Sahiwal, Gir, Ongol, Kangeyam, Vechur. Under sheep, Talacheri is the indigenous breed. Likewise, in dog breeds, Kombai, Rajapalayam, Kaikadai, Gadi Kutha comes under indigenous breed or Desi breed. Moving on to foreign breed, German Shepherd, Golden Retriever, Boxer, Great Dane. Coming to poultry breeds, Asil, Chittagong, Kadaknath, Busra, Brahma comes under indigenous breed. Java, Leghorn, Strahan, Redcap comes under foreign breeds. Moving on to next slide. So in the previous slides we discussed about breeds. What is species? So in lower classes you guys would have studied like kingdom, subkingdom, class, subclass, genus, species. So that species it's very specific to a particular animal. So what is that species? So species is the basic unit of classification and a class taxonomic rank of an organism, as well as a unit of biodiversity. The exchange of genes between the individuals of the species or the gene flow is the major characteristic feature of a species. The gene flow does not occur among the different species. What is animal breeding? So animal breeding is a branch of animal science that deals with the investigation of the genetic value of livestock using different techniques. Animals were selected with superior breeding value including growth rate, egg, meat, milk and wool production. And even people look for certain desired traits. Animal breeding normally is performed to domestic animals to improve desirable qualities and to better suit human needs or requirements. Breeding stock. It is a golden standard protocol for engaging selected animals for the purpose of planned breeding. Why we do this? To achieve valuable traits to bring pure breed or pure line. For example, when you breed a cattle for a milk, so milk is our like a target product, we want more in number. In that case, the breeding stock should be sound, milch, having longer lactation period higher productivity and reproductively efficient. Moving on to the next important factor is environmental factor must be considered or kept in mind, because which will control in breeding stock, which if it is positive end up with reproducible result. If you choose wrongly, which end up with negative result, that means the performance or the product get decrease. Pure breed breeding or pure line breeding, a process of mating animals of the same breed for maintaining pure line. If there is an example of Kangeyam breed is restricted to South Tamil Nadu. If we want that animal in 100% pure line, so we need to breed within that species for several times, where we maintain that pure line for any number of years. So to achieve this, normally people maintain a data where that record is maintained in many dairy industries as well as in beef industries. Sometimes small farmers also maintain this data. Methods of breeding. So under animal breeding, there are two major methods. One is natural mating or natural breeding, which the animal is taken for natural breeding, where some small farmers, they restricted with within their geography, within 5 kilometers radius. So they try to identify the superior male breed within their geography. So which refers mating of animals by natural means, selecting a best bull. Second one is artificial insemination. It is a technique in which sperm cells are collected aseptically from the male and carefully inseminated into female reproductive organ. This process is called artificial insemination. In recent days, 90% of animals being produced through this technique called artificial insemination. Moving on to inbreeding. There are two types of breeds. One is inbreeding, based breeds are chosen. Second one is crossbreeding. So this technique will result with having a different genome or different trait in the wanted animal or in the wanted breed. First we discuss about what is inbreeding. A process of mating of the closely related animals is called inbreeding. This inbreeding is often described as narrowing the genetic base. So within the herd, within a group of animal, again and again the breeding occurs within that herd. If it is poultry, within that group. If it is sheep, within that crowd or within that group. If it is goat, within that group. So the genome or the gene is not mixed from outside, or within the group is not taken to other breed. So within that group, if you maintain that genotype or that particular trait, we call that process is inbreeding. We are not allowing animal to breed with outside breed within the group. Hence we call it inbreeding. Does it have any economical value? Yes. Through inbreeding, we can concentrate desirable trait. Types of inbreeding. Again, inbreeding is divided into two types. Close inbreeding, line inbreeding. What is close breeding? Where animals are very closely related within the family. For example, sire to daughter, son to dam, brother to sister. Likewise, in line breeding, a small difference within cousins, grandparents to grand offerings, half brother to half sister. This occurs only in animals. Disadvantages of inbreeding. There are advantages. Obviously, there is some disadvantages. What are those? Reduced fertility, slower growth rates, greater susceptibility to diseases, higher mortality rates. These are some of the disadvantages of inbreeding. Outbreeding, otherwise called crossbreeding. So crossbreeding involves mating of animals from two different breeds. In the previous slide, we discussed about within the breed, within single breed, we allowed that genotype to mix within that group, whereas in outbreeding, between two breed. Normally, these breeds are chosen that have complementary trait that will enhance offerings, economic value. If you are producing F1 generation, so that F1 generation will have some economically important trait. So superior trait that result in the crossbreed progeny by crossbreeding are called hybrid vigor or heterosis. Again, crossbreeding is like divided into several category based on how many animal you allow for crossing or how many breed that you allow for crossing. So based on the number of breed participate in the mating or breeding, we call that as 2-cross breeding, 3-cross breeding, rotational breeding, composite breeding. We'll discuss one by one. In 2-breed cross or 2-cross breeding, in any form we can call. So under 2-breed cross system, it produces F1 progeny. Normally, like if F1 generation is male, that will be like sold for slaughterhouse or to another commercial breeder. Suppose if the F1 generation is female, then people will adapt that animal to the environment, and that adapted female can be mated to sire of another breed. Because we have chosen two different breed, hence the F1 generation is the progeny or product of first breed. Again, that F1 generation will be allowed to grow to attain like maturity. Once it mature, then probably they allow that animal to breed. So that second breed is with different animal. Moving on to that schematic representation, you can see that this is first breed, we call it as A breed, and second one is B breed. So two different breed which result F1 generation. So this F1 will have traits from both A breed as well as B breed. Now this is a cross breed having both the trait of both the parents, but both the parents are different breeds. That's the important factor that we have to think and plan for further optimization of this protocol. Second one is backcross. It's a well-known technique in genetics. So backcross, in a backcross system, F1 crossbreed progeny are mated to males of one of the parent breed. In the normal system, you can mate with any of the parents, either male or female. It depends on the requirement, whether you need a genotype or the trait from the maternal side or from paternal side. So that will help you to choose which parent, whether with male or female parent, to perform backcrossing. So in diagram you can see like a breed, a B breed. When this cross, the resultant will be F1 progeny. Again, this F1 progeny will cross with either one of this parent. So here we have chosen A parent, where we get again a second progeny, that is F2 generation, where you can see the difference in the genome contribution from the parent or from the mixed genome. So this will help to bring heterosis or to achieve hybrid. Moving on to third technique, 3-breed cross. The prefix 3 different breed. So 3 indicates 3 different breed is chosen or selected to perform this 3-breed cross. I'll show you in the picture. So A is one breed, B is second breed, C is third breed. So in previous technique we have chosen only 2 breed for the crossing, whereas in this technique we are using 3 different breed. So if you use 3 different breed, then the progeny will have triad of A breed, triad of B breed and triad of C breed. Assuming if this progeny receive all 3 triads, may be having a best triad. Probably the animal like the A parent or the B1 or the C, which does not have that superior triad, sometimes F2 generation may get because of crossbreeding. Fourth one is rotational crossing or rotational breeding. Here we will perform a sequence breeding. Rotational breeding is otherwise called sequence breeding, when a male of 2 or more breeds are mated or allowed in this cross breeding. So the F1 generation, again the F2 generation, subsequently F3, F4, F5. So if you test that F5 generation, that will have superior gene system. So we claim that maximum heterosis is achieved by this sequence breeding. You can see the picture. So A and B contributes equal percentage of genome. When F1 and B chosen for breeding were 25 plus 75. Again the progeny is crossed with another A parent, you get 62.5, 37. Again the progeny crossed with another parent, likewise alternate like choosing of the parent, we claim that as sequential breeding or rotational breeding. Sometimes people use only 2 breed, sometimes they use 3 breed for this rotational breeding. So A, second time B, third time C, again A, B, C, likewise, that is called rotational breeding. So by doing this we achieve almost close to 60.35% of heterosis. So this is the best method to achieve maximum heterosis in the breeding system. Most of the dairy industries and beef industries will prefer this method, rotational breeding method, to get or to increase heterosis in their like a required breed. Similar like that of sequential breeding, even this composite breeding is almost same, but thing is we will use different breeds. Instead of 2 breeds, we use more than 5 or 6 breed. You can see in the picture A, B, result F1 generation. Likewise C, D, this combination result F1 generation. So this F1 generation of 2 different progeny crossed, we get again F2 generation. So this F2 generation get mixed to another 2 breed, that is E and F progeny. This E and F progeny crossed with this the hybrid one, which is a resultant of A, B, C, D. So this progeny and this progeny will allowed to cross. You can see there almost 6 different triad this particular F3 generation received. Though we used 6 different animal, but the heterosis is not as equal to the previous technique, sequential breeding. So uh, when compared to sequential breeding, composite breeding can bring heterosis, but not as good as sequential breeding. Grading up. This is also another method to optimize the genome or optimize the wanted triad either in an indigenous breed or in a exotic breed. But in this technique, we have chosen indigenous breed and exotic breed. So with what animal we prefer this particular grading up optimization? We have taken sheep, goat, Talacheri goat and Boyer goat. So Talacheri is restricted to Kerala, Boyer is restricted to South Africa. So what good triad this Boyer has? So you can see that animal size. So this will weigh around 70 to 80 kg. Talacheri breed weigh around maximum 20 kg. So we want this masculine triad in this Talacheri. Why we have chosen Talacheri? There are Kani, Salem Black. There are several species. Why we have chosen this Talacheri to cross with this pure Boyer? So people did several survey, several attempt with different breeds in Tamil Nadu or in Andhra Pradesh, Kerala, even in North India. So when they breed with Boyer, but this Talacheri has given best result when compared to other breeds. There may be almost close to 20 to 25 different breeds, but Talacheri has given maximum output. Hence, they have chosen this Talacheri for grading up. See, what are we going to achieve in this grading up? So, almost this Talacheri genome or the Talacheri triad get retained. In addition to Talacheri triad, we will add Boyer triad to this. What Talacheri triad? Talacheri triad is which will give maximum milk. Another one disease resistant. To this, we will add another triad masculine. But Boyer, it is susceptible for disease, susceptible for drought. If at all we import Boyer from South Africa to India, if you breed or if you raise the animal as in the form of Boyer without crossbreeding or without grading up, the animal will not survive in India because it is specific to geography. This is acclimatized to South Africa. All of a sudden, if you introduce to India, the animal will not survive. Hence, people have selected a best breed to make a cross breed so that this animal can survive even after years together. You can see how the grading up is performed. So in the first generation, where you can see 50% contribution from Talacheri as well as from Boyer. Moving on to F2 generation. So you can see there is in the reduction in the Talacheri genome, and moreover the hybrid genome is keep increased. F2 75, F3 87.5, F4, F6, F7, F8. After 8th generation, almost close to 100%, we achieve that Boyer genome as mixed in Talacheri, almost 100% pure line we achieve. So this is the method we claim grading up, where we are using only 2 breed. So one is indigenous breed, the other one is exotic breed. So in this technique, you can use any 2 breed. If you want that second breed genome supposed to be incorporated to first breed, you have to use this technique called grading up. Thank you for watching this video. We will meet in the next video.